Greetings everyone, this is Yogesh Kalakoti, a PhD student at Delhi. Here I'm presenting a poster that is based on a recent project that we did. It was aimed at building a drug recommendation workflow using transformer embeddings of sequence data. We called it TransDTI, which is a modular framework that incorporates molecular embeddings from various language models and estimates DTIs for a given drug. So advances in sequencing technologies have enabled the collection of huge amount of biological data. So data at such scale have presented a golden opportunity for us to develop powerful sequence-based approaches for modeling protein structure and function. So eventually this would aid the drug development process. So recently you might have heard of AlphaFold, which has basically reinforced the old age adage that all the information that is required to model a protein structure is eventually embedded in its sequence. Uh, so AlphaFold and other such NLP based methods have successfully demonstrated the effectiveness of employing the advances in NLP and by extension machine learning for solving critical problems in bio. With this background, we proposed TransDTI that aimed at employing the rich information that is contained in the sequence embeddings to model the interaction between drug and its target. Coming to the basic methodology that was followed, uh, experimentally validated drug target protein sets was extracted from Kiba, which is a database, and the corresponding smile strings for the drugs and protein sequences were extracted from Gimbal. So these protein sequences and smile sequences were processed using a in-house made framework that employed all of these above listed transformer models to extract the embeddings out of the protein sequences, and similarly there's something called a smile transformer that was used to embed the smile strings into their respective embeddings. Now that we have two sets of embeddings corresponding to proteins and drugs, we use this data to train a multi-level classification model using a feed-forward neural network. The job of the neural network was to uh, predict if a given drug target pair belongs to one of the three classes. The three classes being active, inactive, and intermediate. And optimized a machine learning classifier, we needed a way to validate the claims that the model was making. So we opted for a biophysics-based method that employed molecular docking and dynamics to compare the interaction of a protein with two sets of compounds. The first set of compound was the already known inhibitor for a particular protein, and the second step was a set of compounds that were predicted by the trained model to be an interacting partner of the given protein. So when we did that, if we look at the comparison statistics for both, so red line here indicates the docking score of already known inhibitor with map2k, and in this case the docking score of the known inhibitor with TGFβ. So as we can see, I mean, it's pretty evident that the modular predictions from the transformer based methods, which are shown here in the blue case, does exceptionally well in both the cases for predicting a better lead compound for a given drug target. We even included similar methods from the literature that were recently published for the same task, which are given in red, and it is pretty evident that the transformer based methods did exceedingly well than those. Right. So also the residual level interactions between all the different sets were found to be pretty comparable with each other. Also, we ran a molecular dynamics simulation for all the complexes under consideration, and we found out that the general dynamics of the interactions between the drug and the target for all the different complexes were pretty much similar. So, for instance, the red line here signifies the RMSD for a 100 nanosecond simulation for a known inhibitor with the protein, and the other ones are the ducks that were predicted by the compound, so by the trained model. So as we can see that the profile here is pretty much the similar, and we can see the same for the number of hydrogen bonds that occurred between the drug target pair for all the different complexes under consideration. So to take that further, we wanted to investigate the potential of molecular embeddings that we had just generated. For that, TCine which is nothing but a two-dimensional projection method, much like PCA, was employed to project the embeddings that were generated onto a two-dimensional space. So, to our surprise, we found that the proteins had clustered themselves into protein classes, even though they were never explicitly trained to behave like that. So, this observation demonstrated that uh, the embeddings had the capacity to carry over information so that effective predictive models can be built over top of them. Moving on, these are some data statistics that you might be interested in. So, in conclusion, the study developed an end-to-end scalable framework that can understand the intricate relationships between drug target pairs and make inferences. Moreover, the results obtained reinforces the idea of using representations that try to capture the underlying order in sequential data like protein sequences. So further, the idea of employing structural information to DTI estimation tasks would be an important or would be an important dimension and would be incorporated in a similar pipeline in a future study. So this ends my presentation. Kindly let me know if you have any questions. I would be very happy to